Pollen Viability Assessment. Pollen viability assessment allows to identify infertile male progenitors within the target group. Pollen viability can be estimated by calculating the percentage of viable pollen by means of the dye test with acetocarmine glycerol jelly, or the percentage of germinated pollen in the germination test in vitro. Acetocarmine glycerol jelly test. For the acetocarmine glycerol jelly test, place 1 or 2 drops of 2% acetocarmine glycerol jelly in the center of a slide, previously identified with the progenitor's code. Following that, using a wooden toothpick, take a small amount of pollen out of the capsule and place it on the drop of dye, spreading the pollen with light circular movements. Let it stand for a minute and then put the slide cover on. Mounted slides should lie flat for 1 or 2 days. Afterwards, observe them under a light microscope at 400x magnification. A bright red staining of the cytoplasm of a pollen grain is indicative of viable pollen, while cytoplasm not red or pink indicates non-viable pollen. We recommend the evaluation of 250 pollen grains, expressing the results as a percentage of the viable pollen. A progenitor is considered fertile if more than 50% of the pollen sample is viable. The observation of abnormalities such as tetrads or pollen grains with 4 cores indicates that the pollen sample is infertile. In samples of pollen from diploid potatoes, pollen grains may be larger than normal grains, in which case they correspond to pollen with a non-reduced genetic load. This phenomenon of unreduced gametes is rarely observed in tetraploid potatoes. In vitro pollen germination. In vitro pollen germination is a test that simulates the pollinic tube growth in the stylar tissue, given that the culture medium used in its composition resembles the stigma mucilage. In a beaker of 100 milliliters, add 5 milliliters of a solution of boric acid, 20 grams of sucrose, and 0.2 milliliters of polysorbate 20, commercially known as Twin 20. Complete with 100 milliliters of distilled water and take to a magnetic stirrer to homogenize the solution, which should be at pH 5.5. Once the solution is ready, place 4 drops on the base of a 5.5 centimeter petri dish, distributing them in the vertices of an imaginary square. Then, with the tip of a wooden toothpick, add small amounts of pollen from the same sample on each drop, spreading the pollen with light circular movements. Separately, use the lid of the petri dish to prepare a humidity chamber by placing a filter paper moistened with distilled water covering the bottom of the lid. Cover the bases of the petri dishes with the lids and leave them overnight at a temperature of 20 to 24 degrees Celsius. The next day, place a drop of iodine solution on the sample previously laid on the base of the petri dish and cover it with a 22 by 40 millimeter slide. After that, observe it under a light microscope with 100x magnification. A pollen grain is considered to have successfully germinated when the pollinic tube length reaches a size equal to or greater than the diameter of the pollen grain. Germinated and ungerminated pollen grains count must be performed at a minimum of 10 fields. Results are expressed as the percentage of the total number of pollen grains per field. An average rate of 80% indicates fertile pollen.